Hello everyone, myself Rahul Kumar. I am a third year student of Chemistry Honors in Kironimal College, University of Delhi. Today we discuss about mechanism of enzyme action, taking as an example trypsin. So to begin, let's define what enzymes are. Enzymes are biological catalysts that speed up biochemical reaction without being consumed in the process. They are the essential engines that keep the chemical process of life running smoothly. So how do they actually work? Enzymes function by lowering the activation energy needed for a reaction to occur. By reducing this energy barriers, they allow reaction essential for life, such as digestion and metabolism, to happen at a rate that can sustain life. Without enzyme, these reaction would simply to be too slow. The catalytic power of an enzyme is not spread over the entire molecule. Instead, it is the concentrated around a specific region called the active site. As you can see in the diagram, and this is the diagram. This is the active site, which is divided into two parts. First is the catalytic site, and second is the binding site. Catalytic site, where the catalyst work and produce the products, and the binding side is this where the substrate can bind. Okay, the substrate fit into the specific area on the larger enzyme structure. Let's look closer at the active site. This is the specific region on an enzyme where a substrate binds and the chemical reaction occur. Structurally, the active site is a small three-dimensional pocket or cleft on the enzyme molecules. This pocket is formed by the intricate folding of the polypeptide chain. It provides a unique environment for the reaction to take place. We can divide the function of the active site into two main activities: binding activity and catalytic activity. First, the binding activity. This property increases the building affinity of the substrate toward the enzyme. The active site holds the substrate in position through non-covalent interconnection, such as hydrogen bond, ionic bond, hydrophobic forces, and van der Waals forces. Second is the catalytic activity. This property actually acts in the reaction. It reduces the activation energy to yield a product. This area contains specific amino acid residues that perform the chemical reaction, often acting as proton donor or nucleophiles. Now let's walk through the general step of an enzyme reaction. Step first is the substrate binding. The substrate molecule binds to the active site. This binding is specific and is the result of those nanocovalent interactions we just mentioned, like hydrogen and ionic bonds. The fit is often linked to a lock and key model, though sometimes the enzyme changes shape for a tighter fit known as induced fit. This is a substrate and this is a enzymes where the substrate combined with the enzymes and the formation of the enzyme substrate complex. Step 2 is the formation of the enzyme substrate complex. You can watch here. The, the binding of the substrate creates a temporary intermediate called the enzyme substrate complex. This is the stage where the catalytic action takes place. Moving on to the step 3, catalysis. Inside the complex, the active site lowers the activation energy required to start the reaction. This allows to the substrate to be effectively converted into the product. Some enzymes also use cofactors like metal ion to assist by stabilizing the transition stage or acting as a reactant. Finally, step 4 is the product release. Once the reaction is complete, the product act differently. They have a different shape for the original substrate and no longer fit the active site. Therefore, the products are released. The enzyme then returns to its original shape, ready to be bind with another substrate molecule and repeat the cycle. Now read about the models. So scientists have proposed models to explain this binding process. The first is the lock and key model. In this model, the enzyme's active site is viewed as having a fixed and rigid shape. The substrate fits perfectly into the active site just like a specific key fits into a lock. According to this model, the enzyme does not change its shape, only the correct substrate can bind. The second, more dynamic model is the induced fit model. Here, the enzyme's active site is considered flexible. When the substrate approaches, the enzyme changes its shape slightly to fit the substrate better. This adjustment makes the enzyme substrate complex more stable. In this scenario, the enzyme adapts to the substrate. Now, move the our main topic, that is the trypsin, in which we use trypsin and study about the enzyme action reactions mechanism. Uh, Now that we understand the basic, let's apply this to specific enzyme trypsin. Above things, above mechanism, general mechanism. Try to explain example like specific enzyme like trypsin. Trypsin is a digestive enzyme that breaks down protein into smaller peptides and amino acids in the small intestine. It is produced by the pancreas in an inactive form called trypsinogen. Once activated in the intestine, it cuts peptide chain at a specific point to aid in nutrient absorption. It is also used in medical and industrial application for its ability to cut proteins. Let's look at the mechanism of trypsin in details, starting with substrate binding. Trypsin especially recognizes and binds protein that have lysine or arginine at the cleavage site. These are basic amino acids. They fit perfectly into a specific area called the S1 pocket of trypsin. Inside this pocket, there is a residue called aspartate 189. This residue attracts the positively charged side chain of lysine or arginine. This specific interaction positions the peptide bond exactly in front of the catalytic residues. This brings us to the engine of the enzyme, the catalytic triad. Trypsin uses three specific amino acids: serine 195, histidine 57, and aspartate 102. Here is how they work together. Histidine 57 acts as a base and remove a proton from serine 195. Aspartate 102 helps by stabilizing histidine 57. This process makes serine 195 highly reactive and ready to attack the peptide bonds. Now the reaction begins. Step 3 is the first nucleophilic attack. The activated serine 195 performs a nucleophilic attack on the carbonyl carbon of the peptide bond. This leads to the formation of a tetrahedral oxyanion intermediate. This unstable structure is stabilized by a region in the enzyme called the oxyanion hole. In step 4, this tetrahedral intermediate collapse. This collapse causes the peptide bond to break. The amine part of the peptide leaves the enzyme as the very first product. However, the remaining part of the substrate become attached to the serine 195, forming what we call the acyl enzyme complex. To finish the job, we need water. This is step 5, water activation. A water molecule enters the active site. Histidine 57 acts as a base once again and remove a proton from the water molecules. This converts the water into a strong nucleophilic hydroxide ion OH negative. In step 6, we have the second nucleophilic attack. The newly formed hydroxide ion attacks the carbonyl group of the acyl enzyme. This forms a second tetrahedral intermediate, which is again stabilized by the oxyanion hole. Finally, We reach step 7, release of final product and enzyme regeneration. The second intermediate collapse, breaking the bond between serine 195 and the peptide fragment. The second peptide product is released and serine 195 return to its original form. At this point, trypsin becomes free and is ready to catalyze another reaction. At the end, the trypsin becomes free and catalyze another reaction. Thank you for watching.